Alright, so now that we have looked at the theory of Hardy-Weinberg equilibrium, it's time to actually apply Hardy-Weinberg equilibrium and use that mathematical formula. So Hardy-Weinberg relies heavily on probability theory. And so probability theory is central to understanding evolutionary forces at work in a population. And it's the means of measuring the frequency with which an event occurs. So we have a couple of rules we need to cover. So we have the OR rule, which is the probability that one event will occur or another will occur. Events are mutually exclusive, meaning that both events could not occur. The probability of one event or the other occurring is the sum of the individual events. So for example, looking at a single allele, what is the probability of an AB heterozygote producing an A gamete or a B gamete? So the probability of producing an A gamete is 0.5. The probability of producing a B gamete is 0.5. So the probability of producing an A gamete or a B gamete is the sum of the individual events. 0.5 plus 0.5 equals 1. So the probability of an AB heterozygote producing an A gamete or a B gamete is 1. Now when we're looking at more than two alleles, say, what is the probability of sampling an A or a B or a C allele if the frequency of each allele is as follows: A being 0.7, B 0.2, C 0.1. The probability of either getting an A, B, or C gamete is 1, as it is the sum of the probability of getting an A, B, or C gamete. So pretty straightforward. Now the AND rule is the other rule that we need to pay attention to, and this is the probability that two independent events will both occur. Independent events are those in which one event's outcome does not affect the occurrence of the other. So the probability that both events will occur is equal to the product of the individual events. So again, if we look at a single allele, what is the probability that an AB heterozygote will produce two A gametes? The probability of producing an A gamete is 0.5. The probability of producing a second A gamete is also 0.5. So the probability of producing an A gamete followed by another A gamete is 0.5 times 0.5 equals 0.25. So the probability that these two independent events will both occur is 0.25. Now if we look at more than two alleles, for example, what is the probability that an A allele from a great-grandparent will be present in the great-grandchild? So the probability of the great-grandparent producing an A gamete is 0.5. The probability of the grandparent producing an A gamete is 0.5, and the probability of the parent producing an A gamete is 0.5. So the probability of the great-grandparent producing an A gamete and the grandparent producing an A gamete and the parent producing an A gamete is 0.5 times 0.5 times 0.5 equals 0.125. So the probability that the great-grandchild will have an A allele from the great-grandparent is 0.125. So then how do we calculate expected allele and genotype frequencies? So allele frequencies use an equation called P plus Q equals 1, where P represents the frequency of one allele and Q represents the frequency of another allele. Genotype frequencies are expressed in the equation P squared plus 2PQ plus Q squared equals 1. P squared represents the frequency of a homozygous genotype for one allele. 2PQ represents the frequency of a heterozygous genotype, and Q squared represents the frequency of the other homozygous genotype. Genotype frequencies can be visually related to the Punnett square for a heterozygote cross. So if you look, we have P squared is the AA, so it's a dominant allele and a dominant allele, whereas Q squared is little a, so Q and Q. And then 2PQ is we have two copies of our heterozygous genotype in the chart. So let's look at an example problem. So in a given population of 1,000 individuals, 16% are Cylons, which is a recessive trait. And so we need to calculate the allele frequencies, genotype frequencies, and population distributions for alleles and genotypes for this trait. So our first step is to calculate the allele frequencies. So given that Q squared equals 0.16, we can calculate the frequency of Q by taking the square root of Q squared, which gives us 0.4. We can then plug this into our allele frequency equation of P plus Q equals 1, and the difference will give us the frequency of P, which is 0.6. Now with these allele frequencies, we can then move to the second step of calculating the allele population distributions. So the distribution for the Cylon allele is 0.4 times 1,000 equals 400, whereas the distribution of the normal allele is 0.6 times 1,000, which gives us 600. So that is our gene pool. Now, the third step is to calculate genotype frequencies based off of our findings of the allele frequencies. So frequency of Q squared is given as 0.16. So we need to determine P squared. So we can take P and plug it into this formula of P squared, which gives us 0.6 times 0.6 equals 0.36. Now the frequency of 2PQ can follow the same pattern where we plug in the frequency for P and Q into the equation 2PQ. So we get 2 times 0.6 times 0.4 equals 0.48. Now, just as we did with the allele frequencies, we can take the genotype frequencies and multiply them to our population number to get our population distributions. So the distribution for Cylons is 0.16 times 1,000, which gives us 160 individuals are Cylons. Now the distribution for normal humans is 0.36 times 1,000, which gives us 360 individuals are normal humans. And then the distribution of Cylon sleeper agents is 0.48 times 1,000, which gives us 480 individuals being sleeper agents. So these are essentially the basic methods, and they make some mathematical sense. However, they do not pay attention to the probability rules that dictate them. And furthermore, they do not allow for an assessment of 3 or more alleles. So that brings us to looking at the more standard methods of calculation beyond your intro texts. And these fundamentally rely on the probability rules we outlined, the OR rule and the AND rule. So we're going to take a look at calculating allele frequencies. So allele frequencies are the proportion of an allele in the gene pool. So in a population, there are 2 N genes. N is the number of individuals in the population, and the multiplication by 2 accounts for the fact that each individual has 2 alleles at a given locus. So in a population of 400 people, there would be 800 alleles in the gene pool. So if we're given that there are 100 individuals that are homozygous for allele A, there are 80 individuals who are heterozygous for allele A and B, and there are 220 who are homozygous for allele B. We can then figure out the frequency of P given P represents allele A. So the 100 homozygous for AA individuals have 200 A alleles. The 80 AB heterozygotes have 80 A alleles because they only have one copy of A. So the frequency of P is 2 times 100 plus 80 divided by 800, which gives us a frequency of 0.35. Now Q, which will be our representative of the B allele, can simply be calculated by using our allele frequency formula, P plus Q equals 1, Q being the difference is 0.65. So now we can calculate expected genotype frequencies given the observed allele frequencies. So what is the expected proportion of AA homozygotes? So this is essentially asking us, what is the probability of sampling two alleles from the gene pool that are both the A allele? So this will follow the AND rule. So the probability of sampling an A and of sampling another A is P times P, which equals P squared. Now it's important to keep in mind that this pattern would also follow for calculating the frequencies of Q, or the BB homozygotes. Now, what is the expected proportion of AB heterozygotes? So this calculation involves both the and the or rule. So it's the probability of sampling either an A and a B, or an B and an A. So this is P times Q plus Q times P, which can be simplified into 2PQ. Now, if we're looking at calculating expected genotype frequencies for 3 or more alleles, we need to add another variable to expand our Hardy-Weinberg model. And in this instance, we're going to use R. So the allele frequencies are represented by P plus Q plus R equals 1. So if we look at our genotype frequencies, our homozygotes for the frequency of AA is P squared, for the frequency of BB is Q squared, for the frequency of CC is R squared, C being our third allele. Now heterozygotes have 2 times the allele frequency for the 2 alleles in a particular heterozygote. So we have the frequency of AB individuals, which is 2PQ. We have the frequency of AC individuals, which is 2PR. And we have the frequency of BC individuals, which is 2QR. And what this brings us to is the full mathematical expression is a trinomial P plus Q plus R squared. And if we were to FOIL that, that gives us P squared plus 2PQ plus Q squared plus 2PR plus 2QR plus R squared equals 1. And the importance to keep in mind is we can keep going. So we can add a 4th allele, or a 5th allele, or a 6th allele, and naturally as you get more, you're going to want to incorporate computers into actually calculating this. But it's important to note that using the probability rules instead of the basic methods allows us to calculate frequencies for more than just 2 alleles.